So today we are going to start our notes, and this section is similar in a lot of ways to stuff that you've covered in 7th grade, but we're going to get into a lot more detail about the various cell organelles and what they do, and we'll get into some of the complexity of stuff. We're going to start though by talking about sort of the history of how scientists learned about cells. So what wasn't until the 17th century until scientists really started to learn much about cells. Yeah, it was related to the microscope. So obviously cells are small, and they are so small that you can't really see them without a tool like a microscope. And microscopes weren't really invented until the 1600s. And so once scientists started to develop microscopes, then they were able to look with greater detail at the parts of the cells and start to think about what the different parts do and start to put together some information about cells. So some of the first scientists that researched cells were Anton van Leeuwenhoek. He was a scientist who learned how to make very powerful single lens microscopes. His microscopes were a little weird. They don't look anything like the microscope that you're familiar with. They look like a little sort of panel with a piece of glass embedded in it, a lens, and then a little pin where you place the specimen to be viewed. His single lens microscopes could actually magnify up to 200 times. So he found a way to make these microscopes, but he's very secretive. Other scientists would come to him and want to know how he made these lenses or to buy them from him, but he wouldn't tell them. He wouldn't allow them to use his best microscopes. Turned out that what he was doing is putting little drops of liquid glass, melting glass, letting drops fall into water, and would form these very small spheres, which could give really high levels of magnification. But he didn't really tell anybody about that. He was one of the first people to observe living cells. He looked at blood. He saw sperm cells. He saw living organisms in pond water. And so those were some of the first things that scientists observed as they were looking for cells. Around the same time was Robert Hooke. Robert Hooke, his microscopes looked a lot like what you would be familiar with. He used a compound microscope, a microscope that had two lenses, and that created the magnification. He came up with the term cell. He thought the little compartments that he saw looked like the cells that monks lived in. And so he came up with that word. He published a book that became very popular called Micrographia, where he had drawings of all of these different types of cells that he had observed. And so he showed some of the research he was doing in micrographia. And scientists, studied these cells and looked at lots of different things over the years. Different scientists started to look at different parts of the cells and come to different conclusions. Robert Brown was the person that sort of discovered the nucleus. He saw this structure, and he found it in all of the cells that he looked at. The small structure, a dense structure, he named it the nucleus. Matthias Schleiden in 1839 was a botanist. He was looking at all sorts of different types of plant specimens, and he saw that all of them were made of these tiny cells. Theodor Schwann did the same sort of thing. He looked at all different types of animal specimens under the microscope, and again, saw that all of these animal specimens were also made of these cells. And then finally, Rudolf Virchow in the middle of the 1800s, he postulated the idea that cells actually come from other cells. That they didn't just sort of crystallize like other scientists thought, but that they were actually products of cells reproducing. And so when you put all that together, okay, what you get is the cell theory that had been established around 1855, sort of an overarching idea about cells that these scientists sort of built over the years. And so when we think of cell theory today, we say that it has three parts. The first part is that cells are the basic unit of structure and organization of all organisms. They're sort of the building blocks of life is one way you can say it. That was one important part of the cell theory. Another important part is that all living organisms are made of cells. Some simple organisms are made of a single cell. Other more complex organisms are made of hundreds, thousands, millions, trillions of cells. But everything that's alive is made of cells. And then the third part is about where these cells come from. These cells come from pre-existing cells. So there are some commonalities to all cells. Even though cells come in a huge range of sizes, shapes, complexities, there are some things that they all have in common. All cells have a plasma membrane, an outer layer that separates the cell from its outside environment. All cells are filled with a sort of fluid called cytoplasm, where the parts of the cell are. All cells have ribosomes for making proteins, because cells are composed mostly of proteins. All cells have these ribosomes. And also all cells have DNA, the code for how to build these proteins. Every cell has those four things in common.
What is that?
So when we talk about cells, we sort of split them into two very broad categories: prokaryotic cells and eukaryotic cells. And so prokaryotic cells are relatively simple cells. They are generally small, much smaller than eukaryotic cells. And prokaryotic cells have no membrane-bound organelles. So you wouldn't find chloroplasts or mitochondria or ER or any of the organelles you probably have heard of except for ribosomes in a prokaryote. Prokaryotes have no nucleus. They still have DNA, but it's sort of just spread and floating throughout the cytoplasm. So they're simple cells. We think of them as bacteria or archaea. And here's an example of a prokaryotic cell. They have sometimes a cell wall, a cell membrane, cytoplasm, some DNA that's sort of free-floating in the cytoplasm, and not much complexity besides that. Eukaryotic cells are more complex. They're larger cells. They have many membrane-bound organelles. They might have chloroplasts or mitochondria. They have a nucleus. All of their DNA is surrounded by a nuclear membrane. Now, I think on your notes you might have these reversed. So swap centrioles are a part that is important in cell division. Prokaryotes don't have them. Eukaryotes do. So you might have to switch them.
Okay, so
it should be what's up on the project.
So this one should be absent.
So you'll learn a little bit more about prokaryotes and eukaryotes tomorrow. We're going to do a little bit of reading and make questions about them. And I'll show you this video maybe at the end of class. So as we said, all living things are made of cells. So cells are the basic unit of life. Everything is made of at least one cell. Some organisms are just a cell, like an amoeba, or bacteria. They're just one cell. In more complex organisms, however, like a human, we're going to use as our example. Cells are organized often into groups that complete a certain function. We call them tissues. So here we have a single muscle cell. But in some organisms like humans, those cells work together to form what we call a tissue. So this is some smooth muscle tissue. It's a group of cells all working together to accomplish something. Muscles' function is to contract, to move. So we have the tissue level organization. Some organisms have cells, and they have cells working together to form tissues, but they don't have the next level. Humans do have the next level, which is organs. Several types of tissue can combine and work together to form what we call organs. For example, the stomach. The stomach is an organ. It has some smooth muscle tissue, has some epithelial, sort of skin tissue as well. So it's another level of organization. And then those organs might work together. They form organ systems, like the digestive system is made of many organs working together to accomplish a task, to break down and absorb nutrients and food.
What organs can we live without?
There's lots of organs you could live without. I mean, you can't. Like you could live without one kidney. You could live without an appendix. You could live without one lung. So,
one lung.
Yeah,
Well, can you not breathe as well?
Yeah. But I mean, all those organs, they actually have a function. But you can survive without them. You could live without a gallbladder, right? You could live without eyes, but it doesn't mean they don't have a function. And then eventually, all of these organ systems can combine to form a complete organism. So we call these the different levels of organization. We often will also compare plant and animal cells. There's other types of cells. These aren't the only types of cells. There are, fungi have cells that have some of these characteristics. But often we'll compare plant and animal cells and how are they similar and different. And the similarities and differences are in their structure. Animal cells are missing some of the organelles that plant cells might have. One of those is a cell wall. Plant cells are surrounded by a cell wall made of cellulose, this sort of rigid carbohydrate, polysaccharide. And that's what makes up the cell wall of a plant. Animal cells don't have that. Helps give plant cells sort of their shape and structure. Same thing, chloroplast. Chloroplast is an organelle where photosynthesis takes place. Plant cells have chloroplast, and animal cells don't. Not every plant cell has chloroplast. Cells, for example, in the roots of plants don't have chloroplast because they're not undergoing photosynthesis. The vacuoles in plant cells are different. Plant cells typically have a large central vacuole that's filled with water. It helps build up pressure to push against the cell wall to give plant cells sort of a rigid shape. Those centrioles I just mentioned that are involved in cell reproduction are usually present in animal cells but not plant cells. The shape varies. Plant cells typically have a rigid shape. They have that outer shell of the cell wall, so they have a distinct shape. Often they might be rectangular or have angles to them, whereas animal cells with that outer cell membrane don't really have much of a shape. They're flexible. Also, lysosomes are present in animal cells, but typically not plant cells. So they look a little bit different. You can identify a plant cell versus animal cell looking at some of these structures, looking for them. Today we'll talk just about two parts of the cell. Cell membrane or plasma membrane and the cytoplasm. So the cell membrane, or also it's called the plasma membrane, is common to all cells, whether it's a bacteria or archaea or fungi or animal cell. And it's very complex. The plasma membrane is semi-permeable or selectively permeable. Arpon mentioned this in our question the other day, that some things can move in and out of the cell, other things cannot. Sometimes they can certain times and not others. So transport is what we call this.
Ross, I got a drink going on.
The structure of the cell membrane is called a phospholipid bilayer. Here's an illustration of it. So the plasma membrane is a phospholipid bilayer. What do you think the phospho refers to? It's phosphorus. All this, what you see in blue, these blue spheres, that's the phosphorus portion. Lipid. What is a lipid? A fat. So these two tails that you see that are attached to the phosphorus, that's the lipid portion. And bilayer means there's two of these, two layers. And so there's one layer on top, the outside of this plasma membrane. There's another layer on the inside, and then the lipid is sandwiched between them. Think of it sort of like an Oreo, where the cookie part is the phosphorus on both sides, and then the cream part is the lipids. And then stuck within that phospholipid bilayer, these things like pink, those are proteins. Some of these proteins go all the way through the membrane from one side, and they're on both sides. They're called integral proteins. Some are just on the outer portion or just on the inner portion, called peripheral proteins. Some have carbohydrate chains attached to them. And we'll talk about what these various proteins do. Some of them are receptor molecules. When we talk about receptor molecules that are responsible for communication, those receptor molecules are these proteins that are embedded in the phospholipid bilayer. Now, some molecules can just diffuse through that phospholipid bilayer. Others cannot just go through it. For molecules that can't just diffuse through the membrane, here you see outer layer is the cell membrane. But some of these molecules, small molecules, can be taken into the cell through a process called pinocytosis. So if something can't just diffuse through the cell membrane, it can come into the cell through this process. Basically what happens is the plasma membrane starts pinching in, and these small molecules will be coming with it. And it pinches in until eventually it completely seals off. And then it's sort of a membrane within the cell called a vesicle. And it has some of this fluid trapped in it. That process of pinching in is called pinocytosis. If it's a larger molecule, it might enter the cell through what's called phagocytosis. In phagocytosis, the membrane oozes out around the larger molecule until it's sort of completely engulfed by the membrane. But the end result is really the same. This molecule is now inside of the cell. It's trapped inside of a vesicle, of a membrane, and it's in the cell. Other molecules might travel through proteins, and we'll look at that in a minute. But here's sort of a model of what the cell membrane structure is. So there's the phospholipid bilayer. You can see the phosphates are these gray spheres. The lipids are in the middle. Just two layers. You can see the lipid tails. Also embedded in the phospholipid bilayer are cholesterol molecules. They make the phospholipid bilayer sort of more flexible, depending on how much of the cholesterol is in there. There are these integral proteins which go all the way through the bilayer, with one portion on the outside and one portion on the inside. There are these carbohydrates that are attached to these proteins. There's some of these proteins that are just on the inside or just on the outside. And so this is sort of the complex phospholipid bilayer. It's called the fluid mosaic model. It's fluid because it's flexible. Mosaic because there are these other proteins that are stuck into that. So the plasma membrane, these proteins do several things. Some of them are important to transport. They act sort of as a tunnel. So for example, this transport protein acts as a tunnel where these molecules can diffuse through it. They may not be able to get through the plasma membrane on its own, but they can come through and diffuse through with the help of these sort of tunnels, these transport proteins. Other transport proteins help with active transport, where these red molecules, for example, they're moving from an area of low concentration to high concentration, but it takes some energy to do that. They're pumping this molecule out of the cell. Some of these proteins are enzymes, and they have active sites where the substrate can bind and a reaction can take place, as you see in this middle diagram on top. Some of these proteins act as receptor molecules. That when a molecule on the outside binds to this receptor protein, it can release another molecule inside of the cell, which causes a chain of reactions to take place. It causes some change to happen in the cell. Some of these membrane proteins allow cells to communicate with each other, to recognize other cells. Because of these molecules attached to the outside of one protein, another protein on a separate cell can recognize that and can know what type of cell this is based on these molecules that are attached. This is one of the reasons why white blood cells, for example, won't attack your own body cells, because they can recognize cells based on these attached molecules. Some cells are bonded to, or joined together, and they're joined together by the action of these membrane proteins. Some cells are attached to other external structures, and they're attached to these proteins that are embedded in the surface of the cell. So the plasma membrane has many, many jobs that it does. To look at one in a little bit more detail, this is how cells communicate with each other. So we know that hormones inside of an organism circulate, and then they have effects on certain cells. The cell has a receptor, then these hormones can interact with them. So here you have a water-soluble hormone, a hormone which can't just go through the cell membrane, because we know what's the inside of the cell membrane, what type of molecule? No. What's the inside of the cell membrane?
Oh, the cell nucleus.
So we're talking about a membrane.
Cytoplasm.
The outer layer, the cookie part of the Oreo, is the phosphorus. The middle section is the lipid. Things that are not soluble in lipids can't get through them, because lipids are hydrophobic, and these molecules are not soluble. Instead, however, these water-soluble hormones, they can attach to a receptor protein that has part of it facing outward. And then when it attaches, this receptor protein can then start this chain of reactions, releasing other molecules, which can have some effect on the cell. They might regulate a gene. They might slow down the production of this gene or increase the production of this gene. They may interact with parts of the cytoplasm. But if a hormone is lipid soluble, it doesn't need this protein in the cell membrane. It can diffuse right in through the cell membrane, come into the cytoplasm, and then it can interact with special receptors in the cytoplasm or in the nucleus even, and have some effect there. So it depends on the type of hormone, how it interacts. The second part we'll talk about, again, this is the cytoplasm is present in all cells, whether it's a prokaryote or eukaryote. The cytoplasm is sort of the fluid that fills up the cell. It's not really a liquid, it's sort of like a gel. And the organelles of the cell are sort of suspended within that cytoplasm. Some chemical reactions take place there. It's mostly water, about 80% water. And then there's some other things as well, proteins and lipids and carbs. And that cytoplasm is actually moving. It's called cyclosis, the streaming of cytoplasm within a cell. You can actually see it in this video here. So as the cytoplasm is going through this process of cyclosis in these plant cells, the chloroplasts move as well. And so these moving chloroplasts are just sort of floating around in that cytoplasm. So as you see the chloroplast moving, that's where the cytoplasm is moving as well. So it's not just stuck there, it's not rigid. It's actually moving within the cell. Yeah. So you can sort of slowly see these chloroplasts moving, and that's because they're floating in that cytoplasm. I guess we'll stop with the cytoplasm for now. I have a couple of videos I'll show you. But any questions about the cell membrane or about the cytoplasm?